Hello friends, welcome to my channel Naved Biochemistry Classes. In this video, we will talk about enzyme catalyst hydrolysis in our series of videos on hydrolysis. Enzyme catalyzes hydrolysis of biomolecules, or you can say biopolymers, such as lipids, proteins, and more complex carbohydrates. Enzymes are also called biocatalysts, and it is substrate specific. That is, one enzyme will act on a particular substrate or a particular class of substrate, leading to a particular product. So here it is, you can see the first point: enzyme catalyzes hydrolysis of biomolecules and biopolymers such as protein, lipids, and complex carbohydrate. Enzymes are biocatalysts and are substrate specific. Now, enzymes are also pH dependent. That is, it will depend upon the pH of the medium, whether it is a living system or you are doing in vitro or in vivo experiment. Rather, you can explain like that, and the pH range for optimum activity of enzyme is 5 to 7. You can see that enzyme activity are pH dependent and pH range is 5 to 7. Apart from pH, it is also temperature dependent, and optimum activity of enzyme activity is in the temperature range 298 Kelvin to 310 Kelvin. Okay, so you have enzyme activity are temperature dependent and optimum activity temperature range is 298 to 310 K. On either side, and either you increase it or either it is less than 298, the enzyme activity becomes slow. The enzyme activity on the substrate becomes slow. And as far as human body is concerned, the suitable temperature for enzyme activity in human body is also 310 K. Okay. Now, when you talk enzymes, enzyme has been classified as hydrolase, these are the enzyme which affects hydrolysis of a substrate or of a chemical substances. Now, in a broader term, hydrolysis are class of enzymes which catalyzes hydrolysis, but it is also a special class of transferases enzyme. So hydrolase is itself is a special class of transferase enzyme. You know, when you talk transferase enzyme, you means enzymes which catalyzes, as the name suggests, transfer reactions with water. Okay, with water acting as acceptor for the group transferred. So it is a special class of transferase enzyme, that is hydrolase. Now let us talk some examples. Let us see some examples of enzyme catalyzed hydrolysis. And the most important is hydrolysis of sucrose. You know, C12H22O11 H2O. You have enzyme is invertase, and you get glucose. And fructose, that is glucose and fructose. You must be knowing that this particular reaction is also called inversion of sugar, because originally it is dextrorotatory. The solution is dextrorotatory, but after hydrolysis, the mixture of glucose and fructose is levorotatory. Another one is, you can see, you have pyrophosphate, water, and the enzyme is pyrophosphatase. And what you get phosphate, two, that is, you get phosphate. So you have this, you can see Pi, and here pyrophosphate. Another important is example is, if you have a starch, that is third one, if you have a starch and you have this maltase, you get maltose. Okay, maltose, you can see that it is a disaccharide. Disaccharide of glucose, and obviously it will be C12, H22O11, and you have C6H10O5N, your starch. So obviously you will be having number of maltose unit. The fourth one is obviously, if you take the example of protein, and you have this enzyme protease. Well, this is formed in pancreas, and what you get a mixture of individual amino acid, Similarly, if you have lipid and the enzyme is lipase, and this also is formed in pancreas. What you will get? You will get glycerol, fatty acid, and other products. Okay. If you remove your maltase here, this is the sixth one. It is the starch is also hydrolyzed by amylase. Obviously hydrolysis is going on. In all these cases, you see hydrolysis is going on. The process is hydrolysis. What you get? You get some disaccharides. Mixed product will be, you will be getting disaccharide. You will be also getting trisaccharides. And all the process are examples of hydrolysis by enzyme. So you can see that with the different enzymes, they are going, the different substrate is going hydrolysis, and you can see a starch is going hydrolysis with maltase and amylase, producing different product. So that is the substrate specificity. Okay, and obviously we talked that it is pH dependent, 5 to 7, temperature dependent, 298 to this 310 K, and then we say that we will talk when hydrolysis and enzyme, we talk the class is hydrolase, and which is a special class of transferase enzyme. Thank you.